Yeah, Rakshana, you can start now.
Okay. Okay, sir. Good afternoon.
Okay.
Good afternoon to one and all present here for the IEEE International Webinar Series 73. I'm Rakshana, IEEE student member of EMBS Society from the Department of Electronics and Communication Engineering. On behalf of St. Joseph's Institute of Technology, I'm here to volunteer for today's session. Now, I would like to extend my first and foremost gratitude to our Honorable Chairman, Sir, Dr. B. Babu Manoharan, our Managing Director, Mrs. B. Jessipriya Ma'am, and our Director, Sir, Mr. Sasi Shekhar, and our beloved Principal, Dr. B. Ravichandran, Sir, for giving this opportunity to conduct the webinar. I would also like to extend my gratitude to our Head of Department, Dr. C. Nyanakausalya Ma'am, Staff and Student Affairs, and Dr. G. Rohini Ma'am, Lab Affairs, for the ceaseless support. Now I'll be sharing few words about our speakers. Uh, Firstly, the first speaker of the day is Wesley I. Borisov, who's an associate professor. And the second speaker of the day is Alexei E. Zedanov, who's an assistant professor. And the third speaker of the day is Anton U. Dolganov, who's an associate professor. All the three speakers are from Ural Federal University, Engineering School of Information Technologies, Telecommunication and Control Systems, Ekaterinburg, Russia. Before we start the session, I would like to say few instructions to be followed by the participants. Kindly post your questions in the chat box. There will be a question and answer session at the end of the webinar, and the attendance link will be posted at the end of today's webinar. Now I invite our speaker of the day to kindly start the session.
Thank you very much. I'm glad to welcome the participants of the webinar on behalf of the IEEE Russia-Siberia section. IEEE Russia-Siberia section has been established on 13 February 2003. A section has a strong potential for membership growth through its big cities in the Urals. You can see Ekaterinburg, Ufa, Chelyabinsk, in Siberia, Novosibirsk, Omsk, Irkutsk, and the far east of Russia, for example, Vladivostok, Khabarovsk. The boundaries of our section will stretch more than 5,000 kilometers. The area includes 7 time belts. This year, I was elected chair of the IEEE Siberia section. On the slide, you can see our team of section officers and contacts, how to contact us. I invite to subscribe our Facebook page, Siberian Biomedical Engineering Society. You can see on the slide. I am also the head of the master's program Practical Data Analysis, and I would like to invite you to the master's program at the Ural Federal University. Link on the slide. The program focuses on various applications of data science and data analysis. You will learn how to build artificial intelligence systems using state-of-the-art machine learning algorithms. The program is for software developers and engineers, mathematicians, and computer science specialists who want to move the data science field. The program was implemented in cooperation with SAP company. You will be able to attend several courses from SAP. In addition, you can participate in the research projects at the SAP Next Gen lab at the Ural Federal University. And in addition to theoretical courses, the program includes a project workshop. During the workshop, students are working on real data science projects from companies or science teams. The topics of today's webinar can also be your projects or master thesis. I welcome to apply us, Alexei. Please tell us more about our research project.
Thank you, Mr. Borisov. So let me say a few words about our project. So this project about ophthalmology and bioengineering. So the problem of preserving vision in retinal worldwide. In the evaluation of World Health Organization, around 1 in 3 billion people worldwide live with a variety degrees of visual impairment or diseases. In connection with the growing and aging the population, the risk that more and more people will develop visual impairment or will increase or diseases. The most popular treatment methods it calls conservative treatment, which mainly includes drug therapy. So please, next slide. So before the use of antibacterial drugs, scientists need to test the drugs for toxicity. Ocular toxicity is caused by exposure of retinal tissue to a high concentration of the drug over some time. The retina is also at risk of toxicity due to its rich blood supply. For this reason, it is necessary to effectively assess the toxic effect of drugs. Next slide, please. And no, I think move back. Yeah. No, next slide, yeah. So the methods for toxicity evaluation includes a visual assessment of eyes and electrophysiological methods. So what is it? Electrophysiological testing? It's an assessment of the patient biomedical signals using special complexes or devices. For instance, electroculography includes receiving of biomedical signals from the area near patient eyes, and we may describe the functional states of the eyes based on this such biomedical signal, and especially we may describe the parameters of eyes muscles based on electrooculography. So this principle stay in many applications of biomedical signal analysis. But I have to note that to date there are no well-defined methods to assessment ocular toxicity. This is a topic that our team is working on, which I want to talk about on the next slide. Can you hear me?
Yes, I hear you. Alexei, do you hear us? Sorry. We have some technical troubles.
Yeah, can you hear me?
Yes, sir.
Yes, yes.
Okay, so 1 second. So I should where did I stop? So Okay. So, as I said, to date, there are no well-defined methods for assessment ocular toxicity. So this is the topic that our team is working on, which I will talk on this slide. So the Occulography project is devoted to developing a methodology for the evaluation of ocular toxicity. So this project cooperates with Eye Microsurgery Ekaterinburg Center. So the project can be divided into two parts. So first part is bioengineering part, is the developed of technique for updating, processing, and analyzing of biomedical signals. And the medical part is the processing and analysis of receiving biomedical signal, biomedical information. And the most important part is decision making. Next slide, please. So the project team is scientists from Russia, England, and Romania. Our bioengineering team consists of 2 associate professors and 3 university researchers. Everyone has deep knowledge in biomedical signal analysis and biomedical signal processing, and already done projects with real medical application. So next slide, please. Yeah, and our medical team of the project includes highly qualified doctors of ophthalmology. Especially Viktor Kazakin is the best ophthalmological surgeon in Europe. Last year, he got a golden hand, so he awarded the golden hand. And the second doctor is Mr. Ponomaryov. He is a doctor of medicine, and he is also working in ophthalmological fields. And the third one is Mr. Lizunov. He had a strong knowledge in electrophysiological parts. Next slide, please. So, as I said before, ophthalmologic biomedical signals are not well studied. The tasks of project are recording of this signal, their processing, as well as development of mathematical algorithms for extracting useful information from this.
Sir, excuse me. Sorry to interrupt you. Can you change the position of your camera?
1 second.
Yeah. Yeah. Thank you.
Yeah. So next slide, please. Next slide, please. Yeah. So our aim is to develop a methodology for retinal toxicity, which is very important. So let's imagine how many people have some eyes diseases, and most of these diseases maybe should be developed by use different type of drugs, and of course these drugs can't be tested on humans. So especially when it comes to assessing the toxic effect and any drugs. For this reason, laboratory animals, in particular rabbits, laboratory rabbits, are used to assess toxicity. To test the toxicity, it is necessary to artificially create eyes diseases in a rabbit, after which the antibacterial drugs is injected into one eye and the saline into other eyes. Then it is necessary to record biomedical signals at the set time. After this, we have to administrate these drugs. The animals is observing for 14 days, besides electrophysiological records are carried out by medical staff. So after we use several drugs, we have to record this biomedical signal and analyze these signals to find some progress or regress of our small patient. Next slide, please. So the most significant biomedical ophthalmological signals for assessing toxicity are electroretinography and visual evoked potentials. So electroretinography is an ophthalmic method that can be used to track the state of the light sensitive cell of retina in human and animals. If the visual function is impaired, electroretinography is most often performed. Also, this procedure is carried out to check the functionality of the retina. The retina of the eyes has a large number of pathologies that cannot be detected during the initial examination by ophthalmologist or other doctors. Therefore, electroretinography is performed. The electrode is installed in the special lens of the retina of the eyes, and the signal is recorded by this lens. There is also, we call this multifocal electroretinography. This is the same method, but the electrodes is not one, but whole matrix is small, very small, different electrodes. In other words, we can access the electrical response not from the retina as a whole, but in specific areas of retina. Next slide, please. The second signal is visual evoked potential. It's a method for visual pathway examination from the retina to visual cortex. The stimulation is usually a checkboard pattern that most efficiently test in the central vision. And to implement this method, we need to install 3 electrodes on the back of head, and electrical response will be recorded during checkerboard changing, or pattern board is changing. Thus, the project has many tasks in biomedical signal processing, including work with machine learning methods, which my colleague will talk about. So I'd like to please welcome my colleague, Mr. Dolganov.
Alexei, thank you for introduction. A bit information about myself. I am a young scientist, but already has a position of associate professor. For my 5 years of active participation in scientific programs, I have more than 30 of publication of different level, both journals and conference, that are indexed in Scopus database. I have official degree of candidate of technical sciences, which is somewhat equivalent to a PhD in biomedical engineering. My main 2 topics of interest are biomedical signal processing and machine learning. And on these 2 topics, I have won some scientific grants. And next, I will try to tell you some general principles that we are going to use in these projects. These principles are described on example of my PhD thesis. Every work begins with tasks, and tasks which I tried to solve in my thesis were arterial hypertensions with diagnostics. Then, after we select a task, we need to choose biomedical signals to analyze. In my work, I have analyzed heart rate variability signals, which is time difference between 2 consequent R peaks on the electrocardiogram. Then, after we do a record of a lot of biomedical signals, we need to somehow evaluate different features of these signals. In my case, there were 64 different features, which included some statistical measures like mean, standard deviation, features devoted to distribution of patterns in the signals, and some nonlinear features and spectral features. Next slide. Then, after we create a lot of features, there is some maybe an intuitive task of feature selection. To select among the variety of features we have created, the best features that are suitable for this task. In my study, I have looked from different approaches to find best features, and among the most perspective was approach of genetic programming, when you encode the particular combination of features using, for example, binary encoding. Then you create random population using this encoding, and then you select among this population you have created, you select the best that are suitable for some task. Then you randomly change the best ones, and then you repeat this process again and again, like in natural life. After feature selection, here goes the machine learning. The machine learning, despite its difficulties in its practical application, in idea for me is a simple task, as simple as writing y equals f of x, where x is the data you extract, and y is the expected answer you want to receive from the machine learning algorithm. It could be, for example, classification. In my case, it was classifying between healthy and people with pathology. And no matter how different the particular machine learning algorithm you apply, which may be based on bias theorem or based on closest neighbors or decision tree or some maybe discriminant analysis models, no matter how hard the actual algorithm, the idea is quite simple. Next slide. But when solving some machine learning task, you need to not only find some solution that is good for example, swift diagnostics of arterial hypertension, you need to get to learn how exactly the algorithm decide that this particular person is healthy or not. For that, I needed to revise some basic algorithms that were using for computation, and instead of matrix multiplication, where all the data is mixed, I used some pointwise multiplication, which allowed me to obtain some graphs you can see on the bottom of the slide, which kind of show which particular feature of combination of features among the 15 take the most crucial part in decision making for a particular person. The approach which I described is, yes, based on particular task, but is quite general in a sense that we are going to use the very same principles in our project. So first we will start with recording signals, then we will do feature extraction, then selection, then some particular cases of machine learning, and then analysis of the decision made by machine learning. That's it.
Okay, maybe let's start discussion.
Yes, Rakshana, could you please ask the question?
Thank you, sir, for your stimulating and fruitful session. Sir, shall we move on to the question answer session? So the first question goes like this. What are the applications of machine learning for processing biomedical signals?
The application of machine learning in biomedical signals includes, for example, assisting in decision-making of doctors, so building of some decision support system for classification of some illnesses using different data.
Okay, sir. The next question, why did you specifically choose rabbit as your sample?
Sorry, can you repeat the question?
Yes. Why did you specifically choose rabbit as your sample?
Please unmute yourself, sir. You're not able to do it?
He can, ma. He can.
Yeah. Now,
Yeah.
okay, yeah.
Yes.
So I think I should answer this question. So the using of laboratory animals for estimating of toxicity is this kind of tests has really long history. I think it's from 14th years of last century, and it calls Dre's test. So it was the first test which used for toxicity estimation, and it used laboratory mouses and rabbits. And now, nowadays, the main idea of these methods still stays the same, but some part of these methods had changes. And we cannot, my answer is we cannot use human for toxicity evaluation because of moral principles, and especially FDA and World Health Organization principles. So, and the only way is use laboratory animals, especially rabbits. And the rabbits ocula is roughly similar to the human eyes. So that's several reasons here.
Okay, sir. Thank you. And the third question is, what is electroretinography?
Electro Retinography or what?
Yes, sir. Electroretinography.
Yeah, so this is electrical response of retina of eyes, and we may analyze the functional states of retina based on this signal, and this examination may consist of electroretinography or multifocal electroretinography. In first case, we get the electrical response of whole eye and of whole retina, and the second case is so we may get electrical response of some areas of retina. So, and it looks like the matrix of many electrodes.
Okay, sir. Thank you, sir, for answering all the questions promptly. Now it's time to express my gratitude to all. First and foremost, I thank our guest speakers and thank you, sir, for finding time and giving information on very new technologies despite of your busy schedule. And my heartfelt thanks to our beloved chairman, sir, Dr. B. Babu Manoharan, sir, for this consistent upkeep in conduction of this webinar. And my sincere thanks to our managing director, ma'am, Mrs. B. Jessipriya, and our director, sir, Mr. B. Sasi Shekhar, for their guidance in conduction of the webinar. I would like to express my gratitude to our principal, sir, Dr. P. Ravichandran, for his support to conduct this webinar. I also thank our head of department and IEEE chapter advisor, Dr. C. Nyanakausalya ma'am, staff and student affairs, and Dr. G. Rohini ma'am, IEEE coordinator, head of lab affairs, for their words of encouragement and guidance. And also, I take this opportunity to thank the entire fraternity of St. Joseph's Institute of Technology and St. Joseph's Group of Institutions for making this event memorable success. Finally, I thank all the students, faculties, research scholars from India and other countries for their active participation in this webinar. And our next webinar is by an eminent speaker, Dr. LORENZO MONTE, who will be giving an interesting talk on history of future radar on July 29th at 6 p.m. Interested participant can register in the same link and attend the session. Thank you all.
Thank you, speakers.
Thank you.
Thank you for listening.
Good luck.
Thank you for spending your time and joining with us. Thank you. Thank you.